So I'd like to start off by thanking those who are joining us this evening for the virtual lecture series focusing on our Department of Marine and Environmental Sciences. My name is Brett McAllister. I'm the Director for Graduate Admissions for the Halmos College of Arts and Sciences. With me are two of my staff members as usual, Mr. Theodore Uwe, who, as you in marine scientists, if you are applying for these programs, you'll be speaking a lot with Teddy, as well as Ms. Farah Amro, who I'm sure most of you have been getting emails from regarding our virtual lecture series for the fall semester, which is, of course, faculty members. So any questions that we might have throughout this actual lecture, we'll be having about a 15 to 20-minute Q&A session at the end. So please feel free to type any questions there when we get to that stage. And with that, I'm going to introduce our fantastic faculty lecturer with us this evening. So we have Dr. Fraser Hartley. And tonight's lecture will be on reef fish behavior, ecology, and management in reef socio-ecological systems. After growing up in Africa and the Middle East, Fraser Hartley spent much of his earlier life working in wildlife conservation society in Papua New Guinea and Fiji, working alongside local communities to conserve their reefs and manage their fisheries. This experience led him to a PhD in the ARC Center for Excellence of Coral Reef Studies at James Cook University in Australia, focusing on the interactions between traditional management in the South Pacific, marine protected areas, and fish behavior. Since receiving his PhD in 2014, he has continued to work on coral reef socio-ecological systems with colleagues in the environmental and social sciences during post-doctoral positions at the University of Exeter in the UK and the Institut de Recherche pour le développement in Montpellier, France. Prior to arriving at Nova Southeastern University in September of 2023, Fraser was a Sir Cymru Fellow and lecturer at Swansea University in the UK. His current research continues to focus on reef fish behavior, reef ecosystem function, and reef socio-ecological systems, primarily in the Philippines and Papua New Guinea, but with a recent increased focus on the Florida and the Caribbean. So Dr. Hartley, thank you very much for being here from my staff, and I'm sure from many of the attendees who are here to watch you speak this evening. We're excited to have you. And with that, Dr. Hartley, the floor is yours. Take it away.
Okay, that's a little bit awkward. Anyway, so thanks very much, Brett. I know I threw quite a few slightly strange words at you there in the introduction. So I'm here really today to talk a little bit about reef fish behavior, conservation management. The way I'm going to do this, though, is I'm going to first start off really discussing sort of my journey. As Brett has, mentioned, I've actually been around quite a lot. I've gone to a lot of different countries. I've done a lot of different things here and there. So I started doing an undergraduate degree in marine biology and then my master's in tropical coastal management at Newcastle University. And that really set me off on my way. I was really fortunate that Nick Palunin, who had spent a lot of time in the South Pacific, was one of my main supervisors during that time. And I also met quite a few colleagues there, which I still collaborate with to this day. I had no idea at the time that I'd be collaborating with them still, but we're getting on for like 25 years later and I'm still working with them. The Wildlife Conservation Society has been a really big influence in how I do my work and what I consider science and conservation as well. It actually really started in Kenya, which I'll go into in a little bit more detail in a moment, and then moved to Papua New Guinea. And it really opened my eyes to basically the world beyond ecology and science. And since then, whilst I did really start working on on sort of like marine protected areas and fish behavior, I've become a little bit more of a jack of all trades. I've worked on coral reef carbonate budgets at the University of Exeter. We're now at the Institut de Recherche pour le développement, or IRD, in Montpellier. I worked a lot on expansion of coral reef fisheries. And when I was at Swansea University, I started really looking at mechanisms of behavior as well. But my real journey in coral reef science started with Tim McClanahan and Nyawira Musaga. These are, Tim especially, but Nyawira as well, are basically icons of marine conservation in Africa. Tim is from California originally. He moved to Africa in the 70s. He actually did his PhD in Florida after he came back briefly from Africa. But in 2004, I was fortunate enough to do my master's with Tim and Nyawira. So I was actually in the UK, and they basically hosted me and provided me with a canoe that I could take out to the reef. So I was kayaking like 4 or 5 kilometers each day to my field sites. They put me in touch with local fishermen, which was my first sort of like real experience of looking at the socio-economics of fisheries. And I was looking at how spillover of fish across a marine protected area in Mombasa, which is this area in the middle of the map, influence things like urchin populations and triggerfish populations, and how that changed not only fisheries dynamics, but also the dynamics of the shoreline. But really it was due to Tim. So I did my master's, and then I'd spent a year in Edinburgh basically working in pubs and bars, and not having applied for multiple PhDs because I really wanted to continue being a scientist, and failing at getting any PhD. And I was really a little bit concerned at this. I thought that my journey in marine science was over really before it started. But Tim put a good word in for me with the Wildlife Conservation Society in Papua New Guinea, who were looking for somebody to go out there and basically train people in how to do fish surveys and coral surveys. And that was a really important, I would say, changing point in my life, a tipping point in my life. So I worked for the Wildlife Conservation Society for 3 years, mainly in Manus Province and New Ireland Province. And a lot of this work was training local staff and local people into how to monitor the reefs. And a lot of this work was really doing this kind of top-down, donor-led conservation, where we were basically co-opting local traditional management structures to try and put in no-take marine reserves. Now, no-take marine reserves are something which are really well known in the Western world, and there's quite a lot of evidence and people like them. But in Papua New Guinea and a lot of the South Pacific, where they have these traditional tamboos, the idea of something being no-take forever, effectively, is a little bit contradictory to them. And they also do things which we would not think of as particularly friendly to the reef. So you see here in the bottom left-hand corner, there's this pile of coral. So one of the islands I worked on in Manus, they actually have a monopoly on producing lime from coral, which they then chew with betel nut as a kind of stimulant. And in the top left-hand corner, we have them harvesting a tamboo. So what they will do in a lot of these areas is that they will close off a large area of reef it can be for a few months, it can be for a few years, it can recur, so they do it every year. They might not do it for decades at a time. And it's completely linked with social need. And we're not talking about need for immediate food or food on a daily basis. We're talking really about maybe they need to harvest and get a lot of money quickly for school fees, so they harvest a lot of fish, sell it in the market, they can pay school fees. Maybe a big man in the village has died, they've closed an area out of respect, and a few years later they will open the area, fish it really, really hard, so they have a massive amount of fish for a feast. So these are like just different ways that they were using these traditional areas. And for me, coming from the UK as a 24-year-old who was really focused on conservation and reef fish ecology, and then being confronted with this in Papua New Guinea, where I was living in these villages. Over the 3 years I was there, I lived for a year in these villages. It really taught me to look differently at how you both deal with the ecology of a system and the influence of people on that system, and also whether or not what conservation means and what sustainability means. So just to run through this a little bit more of an example, this particular island is Halmos, where I do a lot of work. It's about 5 square kilometers of reef area around it, but the island itself is less than a square kilometer. And it's idyllic around there on the reef. You've got clownfish, large schools of fish, you've got some great corals, and really healthy reefs, or at least you did when I was working there back in 2007, 2008. But the important thing to remember is that there's also almost 800 people living on that island. It's really densely packed, and they require to use the reef to feed themselves, to earn money, and so on. So they fish it really quite intensively, and they're catching a lot of reef fish. So you can see here in this one sort of picture of a market, you have your parrotfishes, you've got unicorn fish, you have snappers and groupers in here as well. You've got goat fishes, mud crabs, over here in this little corner here, you have various different mangrove mollusks. So they really fish it hard and utilize almost everything that they can from the area. And that means that you actually have some really complicated social structures that have evolved to deal with all of this. So, for example, this is work that I was doing again in just on the island next to it in Andhra, and we were mapping where everybody could fish and what they could do. So it really varies depending as well on what they're doing. So if you're fishing, you pretty much have access to most of the island. But then if you're collecting something that normally lives on the reef surface, even if it's a lobster or anything like that, you can only do it in your clan. And then if you're collecting something which is a traditionally marketed product, so we're thinking here trochus and sea cucumber, then it gets really restricted to your family. But then you have all of these inter-clan and inter-family connections, and it can become a really difficult, really complicated system to manage. So a lot of what I was doing there was trying to understand how people worked and what you could do within a conservation framework that was also acceptable within the framework of the society that lived on those islands. And that led me, so I was there for about 3 or 4 years, and I was really quite happy working in Papua New Guinea. I was working on the islands. I was, spending a lot of time in the villages, a lot of time, a bit of time in the main towns, and I was really quite happy. But whilst I was at Newcastle several years before, this gentleman down in the left-hand corner doing a reef survey, Dr. Nick Graham, I'd made good friends with him, and Josh Thinner, the guy in the top right, had brought Nick and himself and a whole group of investigators to Papua New Guinea, and I facilitated them doing work there. And I had kind of fallen into another rut. So I'd fallen into a rut after Kenya, and then it took Tim McClanahan pushing me and putting a good word in for me. And then it took Nick and Josh, who I'd met, so I'd met them both before, and on this one sort of event in 2009, this one sort of expedition, they really opened my eyes to what I could be doing as well. So Nick is an incredible ecologist. He does so many great things on coral reefs. He's probably one of the biggest brains I know on reefs. Whereas Josh has a great insight into the social structures, and more to the point, into being able to create the theory around that social structures and apply them in the real world. So on this one sort of expedition event in 2009, we did our reef surveys that Nick's doing down in the bottom left. We did fish catch surveys. We worked with the local community to understand their customs and their governance. And this is, on the left-hand side, you see how they will mark an area for tambu. They have a particular type of plant. They plant it on, they take palm leaves, they take a couple of other plants, they tie them together in a particular way, and they put them on the shore to tambu an area, saying that nobody can go and fish in this area. And then we worked with them we worked with other people to try and model various different ways how their environment would change and the decisions that they would make would change based on what we'd learned. And one of the things, whilst we were going through this whole process that came up, was this. I'd lived in Papua New Guinea for 2.5 years at this point. I'd kind of knew this, but I didn't really. And then Josh and I were sitting down, and it's like, they don't close areas. They don't use no-take zones to increase the biomass of fish, which is what we think they do in the Western world. We think that you close an area, the fish aren't fished, they live longer, they grow bigger, you get more fish in there, you get more diversity there. In Papua New Guinea, they close an area to make the fish tamer so that they are easier to catch. And this is something that both of us didn't really think of before this. And we thought, well, that's something that I could do a PhD on. Like, I have the connection from Papua New Guinea. I'm interested in the fishery. And so Nick and Josh, between them, they were both at James Cook University in Australia at this point, effectively forced me to do a PhD here. And I'm really thankful to that, because it led me to a different way of thinking about not only how you can do science, but how you can do applied science. So this brings me to the first topic I want to talk about, which is fishing and fish behavior. So after I'd come from Papua New Guinea, I went to James Cook University in Australia, and I started working on this fishing and fish behavior for my PhD. And one of the important things that we needed to do at first is work out some form of behavior that we can actually measure well. And so we came up with taking an idea from the terrestrial environment, which hadn't really been looked at in the aquatic environment before, and that is flight initiation distance. So flight initiation distance is how close a predator animal can get to a prey animal before the prey animal flees. And the idea is that if a prey animal is experienced, or if a prey animal is particularly wary, they will flee earlier. So that means that under conditions of higher predation pressure, or in this case, if we're applying it to fisheries in Papua New Guinea, under conditions of higher fishing pressure or fishing pressure, the prey will flee earlier in an encounter with the predator, and that is flight initiation distance. Now, that seems a fairly simple concept, but the world in which animals live is really complex, and all sorts of things can influence flight initiation distance and prey flight decisions. And we had to think about what we're actually interested in. And there's a whole host of things. There's crypsis, so how well hidden they are, the amount of camouflage they have, how far away they are from an area where they can be safe from predators, how many other fish or animals there are around them, so how is there safety in numbers. You have the physical condition, how healthy they are and whether they're able to escape, because an ill animal should flee earlier than a healthy animal. But we looked at all of these and we came up with certain things which we thought were relevant to fish and fisheries. So the first one that I want to talk about is size, because obviously in a fishery, you want larger fish, less effort, catch more fish. The prey experience, and this is really important within what I was doing. If you had, this is the whole idea of fish becoming tame and being naive to fishermen. So if you've closed an area for several years, the fish won't know what a fisherman is, and so they'll let the fisherman get close. And then various different aspects of the predator itself. So this predator speed, the predator size. So this is, do fish actually recognize a human as a predator? The directness of attack, so the style of spear fishing, and various other aspects to do it. So in order to be able to do this properly, I ended up having to follow, and for my PhD, I followed spearfishers, took commonalities between the spearfishers in Papua New Guinea, put them all together in one sort of like combined common way of spearfishing that was also repeatable, because every spearfisherman has their own little idiosyncrasies. And you can't do that in a scientific study. You can't just have your own little idiosyncrasies, because they are something that will change depending on the spear fisherman, and certain fish may understand them, certain fish may not. And then we had to first ask this question, because it had never been shown: does fishing actually cause this change in fish behavior? Do fishes become tamer? So this was work that was done in New Ireland province in Papua New Guinea. So I had 4 different sites that ranged from no fishing pressure all the way up to a really high fishing pressure one. This little site beside Kavieng here, that's a really high fishing pressure area. It's right beside the main town of the province. And so I spent effectively a week of my PhD holding my breath whilst pretending to be a fisherman, which was a lot of fun, but also it was very tiring. I was a lot fitter coming out of the PhD than I was going in. And I had various different questions that I wanted to answer that were related to fishing. So will flight initiation distance be larger and bigger fishes? They should have more experience of a fisherman, so they should be able to flee earlier. That FID will be larger if fishes are in larger groups. This is the many eyes hypothesis, which basically states that if you have many people looking out, or many fish looking out, you will see a predator earlier, and then you will leave quicker. So you will leave at a further distance. You won't let the predator get so close. However, in fish, because they have a variety of other mechanisms they do as anti-predator defense within the group, including confusion factor. Lots of fish swimming in various different ways will confuse a predator, and also just the dilution effect. If a predator is coming at 100 fish and you're one of those fish, you're much less likely to be chosen than if the predator is coming at one fish and you're that fish. And then finally, the one that we're really interested in, will flight initiation distance increase with fishing pressure? So what we found for the first 2 questions, that yes, flight initiation distance will be larger and bigger fishes. That fits with the theory of the economic flight hypothesis. Will FID be larger if fishes are in larger groups? We didn't really find that. When it has been looked at in fish prior to this, it had been looked at in like schools of small minnows and guppies and stuff like that, things that were much, much smaller than the fish that I was looking at here. So they were, you know, the minnows and guppies were maybe a couple of inches long. I'm sort of like looking at fish that are over a foot long. And so I investigated these 6 different families that are food fish families there. So we have the surgeonfish in the top left here, parrotfish, goatfish. We have our triggerfish, snappers, and groupers. By the way, if you see a QR code in the corner of the screen, that's actually a link to the paper that was published from this. And were our results. So as you go from the light red to the dark red, you're going from 0 to low to moderate to high fishing pressure. And on the y-axis, you have flight initiation distance in centimeters. So we're going from no flight at all the way up to 5 meters at the top. And what you can notice is that across almost every single group of fish that I looked at, we saw increases as you went from 0 to low to moderate to high. Now, sometimes they were quite step-like. Here in the Scaridae, the parrotfish, you can really see the differences. Sometimes they, such as the Ballistidae, across the 0, low, and moderate, you didn't really see any difference, and then it jumped at the high. Now, that suggests that what's happening for the Ballistidae or the triggerfish is that they aren't being fished under low fishing pressure because there's plenty of other fish to choose from. But as you get to higher fishing pressure and fish become more scarce, fishermen are going to choose these less favored species. So triggerfish can often be quite rough-skinned. They can, you know, especially the bigger ones can be more difficult to spear because literally the spear guns that they use in Papua New Guinea aren't particularly powerful and will bounce off them. So basically the people aren't fishing those triggerfish until they've really fished down their system quite heavily. And if we look, and this sort of orange golden line here, that is the effective range of spear guns in Papua New Guinea. So basically people won't take a shot if they're beyond that range. And as you can see, for a lot of the 0, low, and moderate sort of like fishing pressure areas, most fish are actually below that flight initiation distance. So this is implying 2 things. One, we're definitely seeing an increase, which implies that fish are aware of the predators, but that they are not treating humans as being a particularly dangerous predator sometimes until you get to really high fishing pressures when you have a lot of fishermen in the water and a lot of experience. So that was so that answered the question that this behavior is actually being shown in fish. And now the question is, well, is there a fishery benefit? And to do this work, I worked in Vanuatu. So this is where you had that tambu sort of like sign going up. And I worked across these 2 tambu areas, which were closed for 11 months of the year and then open for 1 month. I had these 2 completely no-take protected areas, and I had 2 areas that were completely open to fishing. And I did a full sort of like spectrum of work here. I did ecological surveys, so I could estimate how much fish biomass there was in each of these sites. I did flight initiation distance behavior experiments, so I pretended I was a fisherman and swam after loads of fish. And I did catch surveys, and I was really fortunate here because I managed to get to this place in Vanuatu just before they were opening up their tambu areas. So I was able to do catch surveys abundance, flight initiation distance, and catch surveys before the tambu event, during the tambu event, and then after the tambu event. So I could really track behavior and whether or not it had an impact on the fishery across this time. So this is what I found. If we look at the left-hand graph at first, again, flight initiation distance on the left-hand side, and we have the no-take reserve, the purely harvested closure or the tambu area, and then the open area. And I'm just showing you the data for the Achidzurdae, for the surgeonfish here, because it probably shows the best sort of like example of what's happening here. And so in the periodically harvested closure, the dark bars are before the tambu was harvested, and the light bars are after the tambu was harvested. First of all, the periodically harvested closure showed a greater flight initiation distance than the no-take reserve, but less than the open. So that implies that a 1 year or less than 1 year closure event does actually change fish behavior. Because immediately after the periodically harvested closure, the tambu area was opened, you saw the flight initiation distance jump from about 280 centimeters all the way up to close to 400. So just 1 week at this point of fishing was really having a big impact on fish behavior. So this was providing a lot of evidence to tell me that yes, this is a real event, and yes, there is a really quick impact of fishing on fish behavior. If you look at the figure on the right, this is looking at the catch per unit effort, or how many kilograms of fish people were catching per hour. And the dark bars in this case are in the open fished areas, and the light bars are during the tambu harvest. What you can see for the Acanthuridae is that effectively you had a tripling of the catch per unit effort just on that one particular family. And when you look at the biomass data, which I haven't shown you here, there is no real difference in Acanthuridae biomass between the no-take reserve and the periodically harvested closure. So that implies that there's something else going on. It's not just that there's more surgeon fish, and so therefore they're more likely to catch them, that there is something else going on in this case. And if you look for all fish, you saw a CPUE of much, much greater, sort of like almost double during the tambu harvest than during regular fishing. And again, we weren't seeing too much differences between the open areas and periodic harvest closure in total biomass. I think there was maybe, the difference between them was maybe 20%, but we were getting a doubling. So a 200% increase in catch per unit effort. So that implies that there is something more going on than there just being more fish to catch. And this was really brought home as well if you look at the differences in the catch between these two areas. So on the left-hand side, you have the catch that you saw mostly in the open fished areas. And these are parrotfish with, so all the green ones are the parrotfish. These red ones are parrotfish too. And the sort of lined one at the bottom, that's a surgeonfish. And this is like the typical surgeonfish, the lined surgeonfish they would catch in this area. But when they opened the tambu, we got a very different set of fish on the right-hand side. You had many more unicorn fish, which tend to grow bigger and beefier. You had larger parrotfish. And you also had, quite interestingly, quite a few large soldierfish and squirrelfish. And that soldierfish and squirrelfish are nocturnal fish, and they're quite easy to catch because they tend not to, flee from you during the day. They just hang out on the coral edges. And we saw hardly any of them being caught in the open areas, mainly because they're so easy to catch that people had effectively fished them down. But in the tambu area, they were still there, they were still plentiful, and we were seeing a very different catch because of that difference in, not just the difference in catch per unit, not just the difference in flight initiation distance, but the difference in what people had decided to fish for. Now, the other thing that I want to point out is that most of the fish on the right-hand side are bigger than the fish on the left-hand side. And we've already established previously that we'd expect greater flight initiation distance in larger fish than small fish. But due to this sort of like difference in the catch, we're not seeing that here. And that again implies that these fish have become tame over the course of 11 months, or more tame than we would expect, because if they were in the open area, there's no way as many large fish would be caught because they would definitely flee beforehand. So that was really the culmination of my PhD. There's a few other bits and pieces I did in there, but those were the main sort of findings. I'm still working on flight initiation distance and fish behavior. I've just actually, the paper that's currently about to come out, I can't really tell you much about the results on that because it's still under embargo, but this is trying to then go beyond flight initiation distance in recreational fisheries and start looking at post-flight responses. So we're looking at the prevalence of post-flight responses, whether if a fish flees, whether it then chooses to be safe afterwards. That's like, fleeing out into open ocean, continuing to swim away from you, or if it only goes, maybe 5 or 6 meters and turns around and rests again. And that's a still vulnerable response. So the idea is that previously I've only looked at going for a fish once and then not going at it again if it fled. But the idea here is that you can maybe do repeated approaches on a fish and see if they're more vulnerable to repeated approaches. And then we've modeled that in across three different areas in the Mediterranean. This is with work with Valeria Sbragaglia in Barcelona. And the idea is to see how much increase in vulnerability you get with increased ability to approach a fish. And then the bottom figure so I have 2 students currently who are working on fish behavior and fish communities. So we have, in the middle, we have Sophia Elrath, and she's working in the Philippines with us, looking at how we can use flight initiation distance on things like compliance at marine protected areas. So Danugan Island, which is where I'm doing a lot of work in the Philippines, it has 3 protected areas and 2 unprotected areas in it. one of those 3 protected areas we know is heavily poached. And the idea is that she's using stereo diver operated video, where you can actually work out using videos how close you're able to get to a fish to identify whether or not the minimum approach distance, which is kind of like flight initiation distance, changes between the fished areas, the unfished areas, and the non-complied with MPA. On the right-hand side, we have Madison Androne. She is a current master's student as well. She is looking at kind of similar things in Florida. We don't have the same sort of protection setup here, but she is looking at changes in the fish community, using doves in the fish community up and down the Florida coast across the seasons, and how that changes depending on spearfishing in the local area. Which brings me nicely now to the next sort of advance I'm doing in my looking at behavior, and that's looking at behavior and reef ecosystem function. So Simon Brandel a few years ago came out with these 8 key processes that happen, these 8 key functions across these 4 processes on coral reefs. And they are things like calcium carbonate dynamics, predator-prey interactions, herbivore-algae interactions, and nutrient cycling. And these functional processes are basically what maintains a reef in a healthy state, or if they start to fall apart, you get the reef going into an unhealthy state. And I'm looking at particularly 2 of them, the herbivore-algae interactions and the predator-prey interactions. So the idea that I have here is I'm looking, because I know that flight initiation distance, or how fearful a fish is, can change across a gradient of fishing pressure, I'm looking at a gradient of fishing pressure here and how the fish behavior is changing and what that means to these functional processes. So the idea in this orange line is that is effectively going to be the number of predators in the system, number of natural predators, I should say. So groupers, sharks, trevallies, and so on. And the idea is that when you've got very little fishing pressure on the left-hand side, you have quite a lot of these predators, and they will inhibit these functional processes by prey fish, so by herbivores and by small predators, because they will basically enact a landscape of fear. These small fish and these small predators, the small predators and the herbivorous fish will not go very far away from shelter. They won't spend much time foraging because they'll be looking out for these predators at all times. And as the predator biomass decreases, the predator abundance goes down. What you should see is this black line is the functional process rate. It should go up. And at some point, it will reach a peak where effectively the predators have been fished down, but human fishermen have not then switched on to fishing the herbivores or the smaller predators. And at that point, because now humans are inputting that sort of fear into the system, you will then see a reduction in those functional processes again. And then MPAs are expected to have different impacts along those different areas. So an MPA sort of here in the middle of the fishing pressure, well, that protects the predators. So we'd expect functional processes of its prey to maybe reduce in their rates, whereas an MPA on the right-hand side excludes the main predator, which is humans, but you still don't have the natural predators really repopulating that. And so you should see an increase in functional processes in those MPAs. And so that's work I'm doing in the Philippines. So these 3 stars are the 3 sites I'm using. It's across a gradient. So on the right-hand side, you have Danugan, which is quite heavily fished, even though it has marine protected areas around it. In the middle star, you have Cagayancillo, where there is a community that fishes there, but it is far enough away from most of the main ports to be relatively low fishing pressure. And then this last star on the left-hand side is Tubataha Marine Reserve, which is an incredibly large marine reserve for the Philippines. It's, like pretty much every other marine reserve in the Philippines could fit into it. It's 3 isolated atolls with huge shark populations, lots of trevally and so on. So it's a really almost intact reef system that I can do this work in. And most of this work I'm now doing with Dr. Andrew Bauman, who is another member of the department here. And we are using various different fish models as predators to try and introduce sort of like these landscapes of fear. And we are videoing basically what's happening to the fish behavior within those. So I've moved on from using flight initiation distance to start to look at things such as fish's use of the seascape around them. This is work I started with Andy back in 2019. We're continuing it now going on it. We've got, as mentioned earlier, Sophia is going to be involved in this. We have another student, Alex Butler, who might be coming out with us to the Philippines next year to do some work that relates to different functional processes on reefs. And potentially we have other projects that might be going on in this realm. We also work with a group in the Philippines Reef and Rainforest Conservation Foundation. They are an NGO that has basically protected Danugan for 23 years now, 24 years now. And we've just signed a memorandum of understanding with them, and with the idea that this will help us get students across there to work on all sorts of different projects. They also have a lot of terrestrial work that they do as well. So potentially there's work that can be done like in the terrestrial biome in the Philippines. So just to show you what it looks like when we actually have some of these down. So in this particular case, we're putting macroalgae down, and we're weighing the macroalgae. We're seeing how long it takes for fish to come in and how much of that macroalgae they'll remove when that macroalgae is down for an hour. So these are unicorn fish. They are the main browser on the reef. There are 3 types of fish that we're looking at. So unicorn fish, the browser, which is the middle one, that is really important for controlling the macroalgae. You have various different surgeonfish. This is just another line surgeonfish here, and they're really important for controlling turf algae. Whereas on the left-hand side, you have this is particularly Chlororis microrhinos, which is a fairly large parrotfish. And what they do is that they scrape and excavate. They will actually take bites out of the substrate, and they remove a lot of different types of algae as well. So the idea is looking at how these 3 different types of herbivores interact on the reef, and how that changes the primary production and herbivory on those reefs. I'm now starting to bring that kind of work to Florida. So I've only been actually at NSU for a year now, so I'm just starting to get this going. And Florida is a really interesting example. So this here on the bottom is basically from 2003 to 2022, I think it was tracking the different groups on Florida's coral reefs. And as you can see here, the biggest group on it was a macroalgae and cyanobacteria, which got to about 25% cover. But I've deliberately cut this graph off because if you zoom out, the main thing that you see on the coral reefs of Florida, the main benthic group, are the algal turf and the sediment. And this is really important. This will be really important because that means that that's what most of the herbivores are grazing on. That's what most of the nutrients that are coming out of the agriculture and cities in Florida is going to probably provide and feed. And so this is really important for nutrient turnover and nutrient flow and flux of the reefs. So really understanding how herbivores are dealing with this large amount of turf and sediment is really going to be really important for understanding not just the ecological process on reefs, but sort of things like how sustainable different fisheries are, how what is the likelihood of recovery from for example, storms or coral disease, because these turfs and sediments can really cover a reef and inhibit anything else taking hold there. I'm also starting to look at what we call secondary production of these reefs in Florida. So this is actually work that's done by another one of my students right now. So one of the things she was interested in was all of the artificial reefs around Florida. And we thought, well, do these artificial reefs actually have the same impact as normal natural reefs? And so we were wanting to look at, well, a lot of these artificial reefs have lots of grunts, snappers, and so on associated with that, which are important predators of what we call secondary production. Secondary production are little larval fishes, they're crabs, and so on. And we wanted to see, well, are there differences in artificial reefs? And then we also asked, how does predator type influence how these different fish are taking up the secondary production? So Lauren Dalton is a student in this case. This is her at work, and this is her setup. So she has fish models. This is a black grouper, and we have a barracuda on the other side. And you can barely see it, but there's a little stick here with a little white circle at the top. That's what we call a squid pod. It is a little bit of dried squid that we've taken out, and what we're looking at is to see how close the fish will get to the predator and how long it will take to get close to the predator to eat the squid pod. So here, this is a really good example of it. What in her preliminary data that she showed me today, very few fish are actually coming right close to the predator. Most of them are going as far away as this porkfish here, which is just, maybe 1.5 meters away from the predator. So there's almost certainly an influence of predator type. Influence of predator on the secondary production. We're still trying to work out whether it's barracuda or whether it is a grouper actually matters. But one thing that Lauren did tell me today was that when she put the grouper model down, she'd quite often get other fish mobbing it to try and scare it away or make it move away. So there's definitely different behavioral questions we can ask within. Okay. I can say more about the social ecological side of it, but I think that I'm starting to run out of time, and I wanted to give 15 minutes for questions. What I want to say though from this though is that if you are wanting to follow a career in science, I had 2 occasions where I pretty much had decided to, the first time I was working in pubs in Edinburgh, and because I had put the effort into my master's, somebody had remembered that and put me forward for a position. And then I was fortunate enough to have good friends who saw potential in me when I was cruising in Papua New Guinea and said, no, you can do more. You've got the brain to do science. And so it's those sort of connections that you make, try and basically maintain them and make sure that, if you have people in your sort of like in the scientific world who believe in you, try and make sure that you still respond to them and give them reason to believe in you, because that does really help in making the next step into science.
Thank you very much, Dr. Hartley. I really do appreciate that. And as you mentioned, we do have a Q&A session available. So for those of you who do have some questions for our faculty with us this evening, feel free to enter them in the chat. And while those are getting going, I like to start this off, particularly when people that do research like you are doing, of course, is, what drew you to this originally, to want to do research in the marine field?
So I grew up in Africa. I grew up, I grew up an hour away from a, sort of like not the Masai Mara, but a really sort of like impressive sort of like terrestrial reserve. Lion, all the big 5 were there: lions, elephants, rhinos, and everything. And so I really wanted to be a zoologist when I first started, like David Attenborough. I really wanted to get into that world. And then I did my first dive in Monterey Bay in California in like 1996, and I was interrupted by a seal whilst I was doing some of my open water skills. And I've really never looked back. I was just, blown away by the ability to interact with the creatures in the world, how close you can get to them. Doing zoology doing work on big cats and so on in Africa is a very different world from marine biology. Marine biology can be so much more accessible. Coral reef science in particular, you can see what you're working on and you can get quite close to it. And it's actually, you're in the top normally 30 feet of water. You can do a lot there, and you can ask a lot of questions and answer a lot of questions.
Awesome. Thank you very much for answering that question as well. So I do have one question that just came in. What have been the advantages of doing marine-related research at NSU, and has your experience been any different here than any other colleges you worked at?
So the big advantage, I would say, for me at least, is this is the first place that I've worked at, even including the places at James Cook University in Australia, where I can go out to my study sites immediately. I can just turn up on a random Tuesday, get on a boat, and go out and do research. That is, to me, is a really big advantage. That freedom, the ability to do your research year round. I've always been in places where I have to go, right, I'm going to plan and go and do a month of research in one particular part of the year. And that kind of limits some of the questions you can ask. It's harder to ask questions about time. It's harder to ask questions about seasonality. And being able to actually set that up here is what I see as a really important thing, particularly when we're starting to talk about behavior, which may change depending on the resources that are available, depending on how many people are fishing and so on. So that to me is probably one of the greatest things I have found here.
Awesome, thank you. Another one just came in. What feedback do grad students have about the field of study there?
what feedback about the fields?
Imagine, so like, I'm assuming the question is based on, the two students you mentioned are working with you. How much, feedback do they get to give you regarding how the research is going, any advice, things like that, I imagine.
So I'll take Lauren Dalton as an example. She's the one who's just finished doing her field work. She and I actually started talking about her field work before she came. She was very interested in doing some diving work, first of all, and also she was the one who came up with the artificial reef idea. She was the one who wanted to try the two different types of predators. Like I was suggesting her using the squid pops and looking at secondary production. So I kind of gave her I would say a larger topic, and she really drilled down on the details. And she's the one who did all of the, she worked out what sites there are. She worked out the sort of like the timetable that she wanted to work around. And for a lot of it, I was more of a facilitator of that. And I also was a person who could point her towards the, I could teach her the theory, I could point her towards the correct theory to talk about the behavioral stuff. And I'm there now that she's writing up to help her with the analysis and so on. I also helped her with some of the field work too.
That's great to hear that our students are getting that true firsthand knowledge. That's fantastic. Next question for you. In your research studying predator-prey relationships, how do you consider variable cognitive ability of fish species and how that might affect response behavior?
Yeah, that is a question. At the moment, I haven't really looked at variable cognitive experiences, but I will just a little anecdote from when I was doing my PhD. So one of the fish families I mentioned are the goatfish. And the goatfish like to lay on top of table corals. And even in the highly fished areas, if you approach them, they would swim away and then they'd come back and sit on that table coral. And I related this anecdote during my PhD confirmation seminar, and I got a fairly high-profile professor going, "Well, fish are just stupid, aren't they? If they do that." And it's like, well, some fish is just habituated and learned behavior. And one of the bits of work I was doing at Swansea, which is still to see the light of the day, is looking at, basically decision making across a spectrum of behavior. So this is within an individual species. So like looking if they display bold type behavior, or if they display sort of like wary behavior, bold or shy behavior, how does that change their decision making when it comes to a predator being present? So I gave them a chance to learn and then looked at how they would make decisions. What I found there, this is data I'm still working out, this is zebrafish, is that if you were on the bold end of the spectrum or on the shy end of the spectrum, so at the extremes, you made pretty good decisions after the first time you were exposed to the predator. If you were in the middle, if you were like the ones that were didn't have either sort of real direction, you tended not to make good decisions until a little bit later. You basically were much more conservative in your behavior, in maintaining the same behavior, whereas the shy and the bold ones would change their behavior.
Thank you very much. Next question. You mentioned earlier in the lecture talking about the different types of equipment that the local fishermen might use and things like that, like the strength of the spear guns that are just kind of different than what you might see in the United States. If those type of things were introduced, how much of a variance do you think you'd start to see?
So that's a good question. It's something which I really want to start looking at. So in Papua New Guinea, they hand carve like the wooden stocks of the spear guns. They have fairly terrible spears, to be honest, that they'll often, they'll grind down themselves. The rubbers that they use on the spear guns tend to be quite frayed and so on. In Vanuatu, which has got more of a French influence and more of an expat influence, I would say, than Papua New Guinea, you see more modern spear guns. And the difference between that I found, because I interviewed people in villages in both Papua New Guinea and Vanuatu, was about 1.5 feet was a difference between those sets of spear guns in terms of the range you get. And at the moment, I'm working with Valerius Bragaglia. One of the questions we've asked is to recreational fishermen in Spain who have, good spear guns. So like, what is the distance at which they would shoot a fish? And they've basically said 4 meters. So we've got this kind of range from, 3.3 meters to 4 meters is a maximum distance at which they would take a shot at the fish. So we do know that there is some difference within the gears, but it's still within a relatively small range. It's not like we're seeing double the distance or anything.
Okay. Couple of questions just came in. What advice do you have for recent undergraduates interested in obtaining a Master's of Marine Science?
I have two pieces of advice for this. The first piece is be sure that it's a Master's of Science that you would like. Like, think about what your career path want to be. If you're interested in doing things such as conservation, environmental consultancy, and so on, a more vocational style Master's would be more useful. Something like the MPS here, for example. If you're really interested in a scientific question, or if you want to, go into academia, then you want to really look at the MS. But it's important to, if you're a recent undergrad, if you can get any kind of volunteer experience, particularly picking up a skill such as, working in aquariums, doing GIS, it's really important to have good data and analytical skills, and it will help you a lot when it comes to your thesis research.
Thank you. Next question. In general, do you find that graduate students tend to enjoy the marine science program?
I haven't had one tell me that they haven't enjoyed it. So I would say that that's pretty much the way it's going. I know that some people struggle more than others, and that can be through a variety of factors. But generally, most people who have got to this point, they only want to do marine science. So they do take the blows with some equanimity and are able to battle through.
That is some good insight as well.
Yeah.
What are some traits or habits you think are valuable as a research scientist?
Don't expect failure and be able to bounce back from it. Your hypothesis may be wrong. You may set out for field work and a hurricane blows through and you can't do it. There are all sorts of setbacks that will get in your way, and being able to meet those blows and shrug them off is really important. The other really important thing I would say is that you need to be friendly and collegiate. It is at the end of the day a collaborative world. You're not there to beat the next person. You're there to work with them so that you can advance science together. So be collegiate, work with people, and look for people to work with.
How did you find your passion in reef fish specifically? One of the students is having trouble kind of deciding what field of marine science they're really interested in.
I ended up going down the reef fish pathway because it was something I was interested more in marine protected areas and sort of like the effects of marine protected areas. And when I was doing my master's research work, the easiest thing to do it on over a relatively short period of time was going to be fish. And then because I did that on fish, I ended up learning a lot about them. I ended up spending a lot of time in the water just observing them. This is something I would say to anybody who's interested in behavior or environmental science, just like spend time observing stuff. You don't have to, be putting up a scientific study, just spend time observing things. And because of that, I ended up getting really interested in their behaviors and you can see personalities in fish. And that really drew me into working with fish.
One of our students, I'll read the question section of it, but one of the students just recently visited Kenya and found it a very eye-opening, type of an experience, as I'm sure it would be. How would you describe the benefits of living in a place like Kenya and understanding the relationship between, people and like the living world?
So I spent time working in Kenya. I didn't really live there for very long, but I think Papua New Guinea and Kenya, a lot of it is fairly similar, is that we are in the Western world, no matter how much we like to go out into the outdoors, we are disconnected from some of the immediacies of living with the natural world, particularly the fact that living on a desert island, living on one of these islands, like I lived on these islands for months at a time with no electricity, no running water, with mosquitoes, with malaria. it's not fun. And people will want to earn money so they can, build a better house, so they can get better well for drinking water, so they can build water collectors and so on. And the way that they can get the money for doing that is to exploit the natural resources. And so so one of the things which I think is really valuable, one of the things that you need to understand is that when they're not doing necessarily the things that you think are appropriate for conservation or appropriate for the natural world, they are working from a different set of values and different sets of needs than you are. And you have to be aware of that when you're working with them.
Thank you. We got time for about one or two more questions, so we do have one right now. I want to make sure that I'm cognizant of your time as well, Professor. I know you got a lot going on. How do you suggest that a student were to choose a research or a professor whose research they're interested in to really commit to, when they're applying to grad school? Like, how do you recommend them get in contact with faculty to discuss things?
Genuinely, so there's two things. One, a lot of us have websites, papers, and so on out there. You can read the abstracts to papers, so you can see what people are, that they're all freely available. So you can see what people are interested in general. And a lot of people, have outward-facing websites that are in more, more of a layperson thing. So do the research, read around them, and then make sure if you're talking to a professor. I receive several, emails a month, which are basically, hi, I'd like to work in your lab. I'm interested in corals. I'm a fish guy. I do some work on corals, but I'm a fish guy. So it's like, make sure that you are asking the professor the right question. If they're interested in fish, but they are a fish behaviorist and not like a fish physiologist, ask about the fish behavior. Do not, it's very much make sure that you are asking the appropriate person the appropriate questions, because otherwise they will they'll quite often just say, well, this person didn't do the work, so they won't necessarily respond to you. In terms of your personal decision about what you're interested in, only you can actually really decide that. But I would, when you're reaching out to professors, ask them where they think the field is going, because it may be that what you're asking is no longer something relevant.
That makes sense. And we can also answer slightly from the admissions side. If you do have questions about the labs that we do offer here at NSU, we have information. If those of you that have already visited, as I recognize some of you that are here with us tonight, have already visited or seen our facility, please do come see the facility, hear about the labs, and we're happy to put you in contact with faculty. Well, that's part of our job in the admissions department, is to make sure you're connecting with people like Dr. Hartley on a good level. That's a big part of it. We do have one more question. This will be our last one. If those who do have other questions would like them answered, please feel free to email any member of my staff. We'll happily pass that along to Dr. Hartley if we can't answer it ourselves. This also might not be entirely your area of expertise, but it is an interesting question, I think. What are the possibilities of becoming a veterinary pharmacist focusing on the rehabilitation of marine mammals, or marine animals, excuse me? Would you say that it can be a tough career, or are there any marine biology specialists focused in the rehabilitation therapy area?
That is completely out of my wheelhouse, I'm afraid. I'm not sure I can really provide a useful answer to that.
Understood. I figured not, just based on what your type of research was, but it's still a good question to have out there. And for that person that asked it, I would recommend just continuing to do your research as Dr. Hartley recommended. Googling around, trying to find what other, what universities are doing for their research, what we might be doing to help kind of set yourself up for a best career. And that will be our last question this evening. So thank those of you very much for attending tonight. Those of you watching later on our YouTube channel or other social medias, please feel free to share this with any of your colleagues, coworkers, friends, other students that might be interested in this field. We're happy to have this information out there. As well, please keep in mind, we do have our open houses coming up in October that we virtually held. So keep an eye out for those, who will send out more updated information as we get a little bit closer. And Professor Hartley, thank you so much for being here this evening. This was fascinating to listen to, as I was very excited to listen. So thank you once again. I appreciate it.
Cheers, Brett.